Our next speaker is Jonathan Barnett. Jonathan's a researcher in computational neuroscience at New York University. I hope I got that correct. In his work with person at the HMI Genelia Research Campus, he carried out a reconstruction in simulation of parts of the motion detection system in the fruit fly, Drosophila, based on physical details obtained from electron microscope images of Drosophila brain and a bunch of other information. Jonathan, please feel free to begin sharing your screen and present anytime you're ready.
All right. So thank you again for that introduction. So my presentation will be based on, like I said, like you said, on the project BATS at Genelia Research Campus on dynamic modeling of extracted neural circuitry during my time with Dr. Lou Sheffer. So essentially, the idea is as a big premise of what's going on at Genelia is one of the projects that I was a part of. Their big term, long-term goal was to take a fly and essentially try to find the connections between neurons and what type of neurons are connected to whom. They essentially get this large-scale structure of the fly brain and hopefully use that for further experimentation on a computer. So essentially what they've been doing for the past few years is they've been taking a fly brain and they've been making slices of the fly over time and taking these taking these slices and imaging it with an electron microscope. From there, they would take the slices of the brain and essentially layer it again to reconstruct the whole neuron again, and where you would find the geometric structures in the computer instead of the actual, and instead of just being in biological detail on like actual viewing it experimentally in a lab. And then obviously from here, then they would take all the neurons that they image and find the connections between the neurons that derive the whole population itself. And so then we have this whole pipeline you can kind of think of where they take a fly and then they they take a fly and they convert the brain onto a computer with all the geometric structures and
lunch break, but it is very short.
all the importances that you would see in all the intricacies that you would see in a fly neuron. So now another question is what about neural dynamics? What about the dynamics that go on in the neuron? And I guess also another thing that I should point out is like, what is dynamics? What are we defining as dynamics and during this project? So one way is to think of an experiment where you have a fly, and when you have that fly, you put an apple behind an object, and essentially you put the fly in some experimental arena, and the fly will obviously walk around and use its olfactory system probably to search the apple. Obviously, during that time, they would you would see neural activity in the fly, and this could be like an example of the neural activity in one single neuron for that one model, for that one fly. And essentially, this, as you see, the voltage is changing over time. It's not always stagnant. It's always has some variation associated with it. This is what we define as dynamics. And so that term that I would say is dynamics in this scope of the presentation is the neuron's voltage change over time is what we denote as dynamics. So from here, obviously you would have a bunch of data of changes in voltages, changes in voltage over time. So what you would do, we'll point out that membrane potential and voltage are essentially the same in this regard. What they would do, a lot of theorists would do, is they would sometimes approximate the data into some type of systems of differential equations or in a simulation. And so then we now have this other pipeline of where we take a fly and we're deriving the dynamics of membrane potential in a fly. So question is when we have these two different pipelines, which is great, we have all this data and all this information. The question is how do we relate the two now? We have geometry of a neuron, and then we have the biophysics of a neuron. So the question is how do these two come together? And this is what the premise of my project was over the summer at Genelia. So the first start out, I'll give you a little background of what we plan to model. The area of the fly that we worked on was the fly visual pathway, where the neurons in this specific pathway are non-spiking. And this is fairly important assumption because it makes the modeling much simpler compared to spiking neurons. And then the premise is that we're not trying to get the exact replica of, so we're not trying to match the voltages and changes in voltage over time with and that data that we would see in an experiment. We're just trying to get the general just a general look of the biophysics that we would see in the fly with some experimental data. So essentially what we're going to do, so what we did was we took this one neuron, and it's the particular geometry so the particular geometry of this one single neuron, and we're going to say that each branch of the neuron is corresponds to one branch in this compressed model. So as you see, we see a little branch coming off the neuron, and then that would be we would denote that as one branch in this one compressed model. So then from here, we would take this one branch and we would give it some type of electrical circuit. We would denote it as electrical circuit where we would have it's very simple electrical circuit. It has different parameters associated with it, where this electrical circuit has an output of voltage. And essentially, going through what each of these terms mean, what C means in this circuit is C is capacitance. And how you can think of C is in this electrical circuit, the electrical circuit is receiving current either from another neuron or from an outside stimuli. And essentially, what happens is that when you input large so if you have a one current value, when the capacitance is large, the neuron feels less of an effect from that current. But when the capacitance is smaller, the neuron feels larger fluctuations of voltage based on that one current value. For EL is what we think of leakage, and how you can think of this is that if a neuron wasn't receiving any outside input or outside stimuli, and it was just sitting there, where would the voltage sit at? And in this case, in this model, it would be sitting at -65 millivolts. Finally, we have the value of R in our cytoplasmic resistance, which is RCYT. This is telling you how much does the membrane potential change. So if you were to input a current into the neuron, how quickly does the membrane, how quickly does the voltage change, essentially? So with large resistance, it's much slower change in voltage, while smaller R values would have more rapid changes in voltage. So then from here, we would take each of the circuits and we would connect them together for each branch. And essentially, how you can think of it is that one neuron has probably, I would say, around 200 of these circuits, probably more or less. And it's a very broad and large circuit that we have to simulate for this model. And from here, from here, then we would give the circuit, so this is what the biophysics or dynamics that would look like for that one for that one circuit. And as you see, below is the current. So we have current over time, and current is something that we're inserting into the neuron. And then we have memory potential, which is changing over time, or voltage. And essentially, as you see, when we insert current, the memory potential increases, but it doesn't increase right away because of that R value. The R value is feeling slightly slowing, making sure that the memory potential doesn't just skyrocket straight to the value that the current's inserted. And then when you turn off the current, the memory potential slowly decreases over time back to what is EL. So as you tell, like when we don't insert any current, when current's 0, the neuron is just sitting at this steady state where it's not, where it's around like negative 65 millivolts. From there, if you can insert negative current, then we see the voltage decrease over time. And then it'll increase. Same idea as positive current, but instead this is going the other way around. So in that past one, the dynamics for the circuit was very simple. It wasn't much complicated, but where the most of the complexity into this model, where is the most complexity coming from, is that majority of this complexity is coming from the synapses, which is the connection between the neurons. And when when two different neurons are interacting with each other, we see a more complicated type of membrane potential, where the membrane potential is not just simply moving up and down. It has, like, for example, this skewed Gaussian, or like this hill that's skewed a little bit to the side, skewed from the center. And this is where majority of the complexity of the model is coming from. It's mostly coming from the dynamics between neurons, the connections between neurons. So essentially, what we'll do is that, like, we would connect all the neurons together. So this dotted line is denoting the synaptic connection between neurons. And the way we would connect the neurons is, in the Connectome data set, there would be a large data set telling you where would each neuron be connected to on which branch. So it'll give you like a general neighborhood of where the connections were, where each neuron was connected to. And essentially, what we would do is that we would just find the shortest path between two branches. So for example, in the data at the Connectome, we would see, for example, these two branches that you see here connected. There would be like an ID on that telling you, like, oh, this branch has a connection and it's connected to the neuron below. And essentially, what we'll do is that we're gonna find the shortest path between those two branches, and essentially that would be a connection between neurons. So from here, we would connect a whole population of neurons, and then we would have this whole section of the flybrain that has both a geometry and so it has the geometry and the biophysics associated with it. So this is great, but now another question is how do we test the responses of the model? So now we're curious, is like, now we have this whole model that we have for one section of the flybrain. What can we do with the model? What is one test that we can use to just see how the model fly responds to these input stimuli? So what we decided to do is we decided to use a motion selectivity experiment. And the idea of to get an idea of what motion selectivity is that you can think of you having 3 light bulbs, where I would light the light bulbs up in a sequence, and currently they're all off. So for example, you see a light bulb lighting up, and the light bulbs lighting up in this sequence. As you can tell, if you were while you were watching that, you would think that the light is moving from left to right, when obviously in some regards, like it's not really moving left to right. It's just lighting up and off in just some specific sequence. But this is some part of the fly brain is able to detect this, and the fly brain that we happen to have the model of has this part of that fly was part of this motion detection system. So what we did is that we now have this model fly where this is on the computer now. This is where we made that whole structure of the neurons and the biophysics associated. We did the same thing, and we introduced the lights to the fly again, that specific sequence. And obviously we'll see like as the fly can see left or right. So now what we're going to do is we're going to return back to this model, and we're going to simulate it. And we're going to notice that we received the dynamics of this one particular population of neurons, which is considered in the T4 section. And I would say that each line that you see in this this plot in this compressed T4 right-left stimuli, that each one of those lines is the membrane or voltage of one single neuron over time. So as you see, the activity of the neurons when I first introduce the stimuli, they fluctuate rapidly on the membrane potential, but then they slowly die off. And so now we have that model. But the question is that when we did this one particular simulation, usually the simulation time would take around 1,000 milliseconds. Simulation duration was in itself quite long, because if you kind of think about it, like you see here is 20 minutes, but the thing is that as we move forward, this is really this is a really short time for simulation. It's only like essentially 1 second, and it took 20 minutes to take, and it was also a very small neural population. So the question is like, this is great, but is there a way that we can go simpler? Can we make the model simpler so we, where we're just concerned with just qualitative behavior of the neuron, can we make a simpler model just to understand what's going on? So the way we're going to do this is that when we made this model, we made a simplification where we took each branch and it's corresponding to one branch in a dendrite. So the idea is that why don't we, instead of go even simpler by taking this whole complicated structure in neuron, just simulate just as if it was just one neuron, and then how? So the question is, how would we get there? So the idea of how we would do that is that we made a simplification by saying, like, we're just going to take the compressed model and we're going to compress it even more into this one single node model. And essentially is that we take, so then we do the same idea. We're going to take that complicated geometric structure, make the simplification of making each branch part of, as a branch of the compressed model, and derive the circuit again for that. And from here, the idea is now the way we would move between the two to simplify is we're going to take the capacitance of the neuron and all the capacitances of each compartment. So in this circuit, there are 2 compartments in this, and we're going to also take all the resistance of that, and we're just going to add up all the areas and multiply them by the capacitance and area and also resistance to derive this new capacitance, which is so we have like a capacitance C hat and a resistance C hat. So we have these 2 new values that we derived from adding up all the areas together into this one neural model. And what we're going to do is that we're going to now just do one circuit for that one neuron. So this whole circuit is just for one neuron now. And essentially, that circuit though would just have some renormalized capacitance value and some renormalized resistance value. And essentially, these would be this would just be an extremely simplified model of the neuron. And essentially, one thing So from here, then we move between these two different circuits, and definitely would point out that there is no changes in the synapses. Each neuron will receive the same amount of synapses. They would receive the same amount of the same differential equations associated with them earlier. So again, majority of the complexity is coming from the synapses themselves. So now we have this whole new method where we go from geometry to this compressed model to all the way down to this one single neuron. And we're going to run the experiment again and notice and run through the synapses. So the dashed line is synapses between neurons. And as you see, so again, this is what the voltage looks like, and the same type of neural population with the same type of stimuli. And each line is the change in membrane potential for a single neuron again. So this is just a simple stimuli from right to left. And essentially, we're going to return back here and notice that we had this one for this much more complicated intrinsic model. And as you see, when we compare the two experiments, they look qualitatively very similar. It's very close to each other in terms of look. And in terms of quantitatively, the changes between the two, the changes in membrane potential is really small. And I will point out that the scale that is really, really small. It's like negative 0.02 change in voltage. And this is also really great because then we can look at the time average of the simulation duration. So in the red one, in the compressed model, it was when you ran a simulation of it for 1,000 milliseconds or 1 second, it took about 20 minutes. But when we did it for 1,000 milliseconds for this simplified node model, it only took half a minute, essentially. And this is great because now we have a quicker way to figure out what does the qualitative behavior looks like for a single neuron. So essentially, now a question comes up is like, what can we learn from this and from these results? And essentially, it seems like a big importance is placed on the actual connection between neurons, because if it appears that a majority of the complex dynamics between neurons was based on the synapses. The neuron circuit itself, or the circuit for a single compartment or single neuron, was very simple compared to synapse. And it seems like there's a lot of computation involved between two different neurons. And it also, when we can think of it, is that when we think about extrinsic versus intrinsic properties, it seems that intrinsic properties play a big role in it. Really defines how the dynamics are. In addition to just intrinsic properties, these two play some type of have some type of aspect with each other, because obviously bigger neurons are going to affect how the capacitance changes and resistance changes. So there is an interplay between extrinsic and intrinsic, but it seems like there's a larger importance played on intrinsic properties of a neuron. Finally, in terms of the experiment's result, it seems like that the population structure and the neuron geometry, it seems like there's a greater emphasis on how the population is structured, how neurons are connected with each other, and how that leads to different computation, for example, the motion selectivity experiment. Because obviously, if you had different synaptic structures in a population of neurons, how would that affect the population dynamics as a whole? So in terms of future directions, one of the future directions is the differential equation model. And essentially, a lot of this is playing into the aspect of synaptic modeling. And one important aspect is the idea of neurotransmitters and receptors, because neurotransmitters and receptors each have a specific model associated with it. And the idea is that another question is like, how would we model these synaptic modeling for neurons that that are not spiking? Because the modeling is very different between spiking and non-spiking in terms of synaptic connections, and this definitely plays a huge role in. There's in a lot of future directions. This is something that we have to think about in terms of when we synaptic model each neuron. In addition to that, synapses also have some type of internal state. You also have to think about the aspects that probability does play a role in synapses, because essentially communication between neurons with synapses, there's a chemistry involved, and obviously there are some elements of probability involved when you have exchanging between different chemical compounds. So there's also probability that we have to also think about. Finally, and more exciting things about the Connectome project in terms of neuron geometry is that so a lot of the things that is going on is, one big thing that's coming up is the central part of fly brain is expected to be done pretty soon, and that is quite exciting that we're getting closer and closer each year to figuring out how to upload this whole having this whole fly brain uploaded onto a computer. And essence also, the whole fly brain, male fly brain with ventral nerve cortex project that they're slowly moving towards. It's a current project. And when they finish that, then they would then work on the whole female fly brain with the ventral nerve cord. And then after that, possible to have now whole-scale, large-scale simulations of everything going on in that one in these fly brains, which is quite exciting to think about in the near future. So from there, I would definitely want to say thank you for obviously giving me the opportunity to speak at this workshop. It's been quite an opportunity, and I definitely want to thank Dr. Lou Sheffer for mentoring me during the summer at Genelia with this project. It has been quite an experience down there, and also Genelia Research Campus as a whole has been a great opportunity to explore the Connectome project with them, and most especially in Dr. Lou Sheffer's lab. So thank you.
Yeah, thank you very much. So we're going to have about 50 minutes of questions, and then after that, we can continue with questions if we're all still feeling up to it. And if there are questions, we'll have a little break between talks. I'd like to ask, first of all, if there are questions here in the audience. I'm a little surprised because I have 12. Okay.
Yeah, I was going to say we have some backup slides here on what it is we had to assume when we simulated the Connectome is what we knew. And so I thought that would be interesting to this audience. So if we have time left over, we can go into that. Did you append those to the presentation, Jonathan?
What again?
I sent some slides on what we know versus what we had to assume.
Yes. So like, yeah, I tried summarizing general idea of what's going on with that. So yes, I brought up the current findings for the Connectome. There's obviously different aspects of going on with figuring out how the synapses work, especially one big thing also is how do we understand plasticity and between two different neurons, because obviously when we're trying to image these neurons, it's not very easy to just because since we cut up the fly brain, it obviously can't make new synaptic connections, so it's very hard to find out changes in plasticity in a neuron when we are just looking at this uploaded geometry of the neuron. So there's obviously new things that we need, new methods that we have to use in order to figure out how learning may work and for a fly's olfactory system or plasticity at large.
So did you have slides on that you wanted to show? Because I'm I'm interested in these assumptions, and that might actually take away some of the questions that I had. And I don't know if you
Yeah,
guys-
I can definitely. I definitely have the slides with me on there, so I will pull them up. So. So here, I'm pulling it up right now. So here's some extra info that was given. So obviously, some additional information is also obviously the information of gap junctions. So that definitely plays a role in like non-spiking neurons. And then I brought up like the idea of like neurotransmitters and receptors. And the idea is that there's different methods for figuring out neurotransmitters and different methods for figuring out the receptors. So I was wondering if you guys have any questions on that.
For the one we had here, we were lucky because this is a circuit that's been studied for a long time. And so people had already done antibody staining, and we knew which synapses were, which neurotransmitter to associate with every cell. Now, if you were doing a whole brain, you wouldn't have that for most of the cells. But that's one of the reasons for trying this first in the optic lobe, is this has been studied for many years. And so we knew some of this information. Receptors, we had to guess too, because it's you can't see a receptor on an EM image. And so, and worse yet, the same transmitter can actually have some receptors that excite and some receptors that depress. And so you don't even necessarily know the sign of the connection. But in this case, we did, because people have been modeling motion detection for many years.
Could you identify the receptors, see which ones were receiving the transmitter as inhibitory or excitatory? Or is that impossible?
No, people can do that, but they have to like stick the electrode in the cell and then measure it.
That's electrode recordings.
Yeah,
That's not from the image itself.
Yeah.
Okay.
Did you see what it was? Another question on the YouTube channel?
This is Ken. I was just going to ask, you didn't feel any need to put any voltage gated ion channels. I don't know much about those neurons, but I was a little surprised that there weren't any voltage gated ion channels that needed to be modeled.
I mean, the idea in this model, I guess, I'm just talking about more on the aspect of the synapses, correct? Or the in terms of like, I guess, I don't have enough background to know about voltage gated channels. So
if you could say something about the electrophysiology data that that is known for those particular types of cells, like are they mostly passive when you inject current into them?
Yes, because this definitely was a big assumption going into the model that each neuron was just a passive neuron. There was no spiking involved. There was no like fast changes and rapid changes in voltage.
Thanks.
Yeah, these ones are, if you're trying to model the spiking cells, you absolutely need the voltage gated channels. But these ones are known to be graded. Brighter light, higher voltage, that's right, all the way through. So we didn't feel compelled to use those models. Also, we weren't trying to get millivolt by millivolt accuracy versus the actual measurements. We were just trying to show that if you did left to right motion versus right to left motion, it would trigger the left to right motion cell more than the opposite direction of motion. So we're just trying to get the behavior qualitatively correct and not quantitatively correct on the level of millivolts, which probably would require the channels that Ken's talking about.
I'd like to ask a question that takes into account something that just showed up on the YouTube channel where someone was asking about, say, this is kind of brain of Drosophila, taking the motion network for that, taking those cells, and then it's a generic type of cell. We're using all this information that's already been procured from electrophysiology and building. Simplification also, which was an interesting part that Jonathan showed. And then, so what about differences between Drosophila brains? And I think that ties into a main question that, I could put this question differently. I could say, since we already have the electrophysiology of these motion-sensitive cells in Drosophila, and we can already make a model of them. In fact, we can make simplified models of them if we want to, and we can tie them together, and then we can show how they react to the motion. What are you getting extra from looking at the EM image? What are you getting by looking at the connectome that we didn't already know from the electrophysiology? And either of you could answer.
I would definitely say that, I don't know, would you like to go, Lou, or should I explain?
You go first.
Okay, so I would definitely say that the simplification, you definitely still needed the geometry because you still needed the size of the neuron, because that was a way how we simplified it. Because what we were doing is that when we moved from the capacitance, I can actually return to that slide to make it easier. When we moved from this circuit to the other one, that we also needed to know how many compartments there were, and also the area of that one compartment. So like obviously there is an area associated with this compartment, an area associated with this, and we don't know this information essentially until we know the actual size of the neuron itself.
I would say from a more philosophical point of view, there are many different motion detector models, and we didn't really know which one the fly uses. And so until you get the connectome, then we said, okay, we think this is what the fly uses. But it turns out that even this was not enough. There was another one done by Srini, where we took the network we got from the connectome, and without any bias said, can I adjust the weights? Make this a motion detector. Can I adjust the synapse weights? And he found it was ambiguous. There were actually many ways it could, even the same network could work to be a motion detector. And so without the synapse information, we wouldn't really know how the fly does it. You're right, we have a model, and it tells us what the fly what the fly detects when you go left or right, but it didn't say how. And so this kind of simulation is needed to say, okay, this is how the fly actually does it, versus one of the many ways it might do it. It's like looking at a computer. Right, that program could be written in zillions of different ways to give you exactly the same thing on the screen. The only way you can ever know is to look inside and see what it's actually doing.
By the way, I wanted to invite any of the other speakers who are online on Hangouts to ask questions directly if they like. Since you're already there, you just have to unmute and you can ask anything you want. Ken already did that once, and any more questions are also welcome.
Well, can I ask another one?
Yeah.
So what do you think is the next circuit that might be tackled in Drosophila in terms of really showing that you can connect structure to function with connectome data?
So I think that's more of a question for you, Lou.
Okay.
Yes.
All right. I would hope the next one would be the olfactory memory in the mushroom body, because that's the first place we have any memory, really. And so we have the electron microscope reconstruction. We have a number of measurements. There's a guy who's done a lot of measurements on how it learns smell. Do you have that one picture I sent you, Jonathan, that shows the smell?
Yeah. Yes. Was it the same? This one?
There's only one picture I sent, I think.
I never received that picture.
Okay.
Fortunately.
All right. I said, is somebody here and see if they can.
Yeah, we could try to.
Or I could draw it on the board here. Is there a camera?
Yeah, go ahead.
Yeah. So in the flies, Can you turn the camera around so that people can see it on?
It's already turning to you.
Okay, all right.
That's working on
All right.
this.
We have in the smell system, there's a output neuron, and you present a new smell, and the thing looks like this. It goes zzz, and gives you a nice spiking behavior during the whole duration of the smell that you present, and then it goes flat again. Then you can take the same fly, you pair the same smell with a shock, and then afterwards, once you've done that, now you present just the smell alone again. And the next time you present it, you'll see some waveform that goes like this. Where, in this case, the cell was saying, you know, okay or neutral, I guess. In this case, it's saying, oh, that's not so great. And for this one, we actually have a good idea of what the circuit is, and we think we know how it works. And the way it works is the smell comes in. There's about 60 different receptors for different things, 60 different chemicals that it can detect. On the average, about half of these is turned on. And that goes through sort of a scrambling network. And out comes about 2,000 neurons. And in here, there's a sparse coding. In other words, typically only 6% of these are turned on. That's a different 6%. It's almost like a hash code if you're familiar with computer science and stuff like that. Then there's a memory system where these ones tie into these output neurons. And that's what we're measuring here. And that's measuring giving some sort of output. Like in this case, it says, okay, when this smell came in, I'm going to turn on. And then coming in here also are another signal going in that says either good or bad, either reward or punishment. And then when the signal comes in at the same time the smell comes in, then these synapses at the intersections here are strengthened or weakened. And then the next time the same smell comes in, you get this different response over here that either attracts or repels the fly from that. And so we have, for this part here, we have this whole section. We have a electron microscope reconstruction of this part. This is how we think it works. What we don't have is a really good model of how these synapses become stronger or weaker when the good or the bad effect happens, and how that, we know it's at least partly on the presynapse side, but we don't know much else about it. We know it's on the presynapse side because if we suppress the firing in these cells, it still learns. And since we suppress the firing, the output never happened. Right? This thing over here was never triggered, but yet it still learns. And so we know at least some of the learning happens on the presynapse side. But it's not at all clear whether it's all there, or there's some on the postsynapse side, or how the circuit works. And so I would think this would be a natural next step from the one that we got, if that's the answer to your question. And someone, I think, asked also, what about the difference between different flies? These flies are really hardwired. A fly like in the optic system, the part we demonstrated, the fly needs that optic part to fly. And you can take a fly, you can raise it completely in the dark, so it's never seen a single photon in its life. When you let it out as an adult, it can fly just fine using that motion detector that you saw. So that part wasn't learned. That part is really, really hardwired. And we've done, in this case, a couple different flies. And every fly we looked at, that circuit is very nearly the same. It has variation between the different columns on the fly's eyes. I got this big compound. So you see variations between the columns. And you see variations fly to fly, but they're relatively small. These things are really, really like little hardwired machines.
But then again, what you're pointing to right now, the olfactory system, is where you get differences because of learning, and that's what has to pop out.
This seems like the two extremes. Like it seems like when a fly is born, it can see perfectly well, and that part's completely hardwired. Whereas the olfactory system is the opposite. It's a blank slate when it's born, and it learns based on whether it finds reward or punishment associated with different smells. That's not quite true. There are some smells that go straight into the brain. The sex pheromones and stuff like that are hardwired. It doesn't have to learn them. Whereas this approach or avoid appears, as far as we tell, to be completely learned.
Did that answer your question, Ken?
Absolutely. Thank you.
Awesome. This also immediately addressed some of my questions about testing differences in learning I had one more question about the results, the things that the conclusions you drew, where you were saying, well, if we look at the difference between how much do the intrinsic or intrinsic properties, and whether they matter, are populations versus individual neurons. And I'm wondering, when you determine these differences, so what would you say is the reason, for example, that the extrinsic individual neuron structure properties play less of a role? And if you had to sort of theorize, like, how does the Drosophila brain go about making that so? Or is this just kind of something that pops out of doing the simulation, and there's no like built-in constraining factor or something?
And so I'm guessing you're saying, how is extrinsic less important or more important than intrinsic?
Well, that was just one example. I think I'll have to rephrase this. When I think about, say, human brain function, very often, I try to also think about how can one area in the brain even ever hope to understand another part of the brain. We say everything is so different from one place to another, and there's, the synapses are probabilistic. Sometimes they'll fire, sometimes they won't fire. You'll never get the same spike train in a row. All these things, and yet, the brain can understand itself. Different areas are talking to one another. So I often think about, how does the brain make sure that some parts of what's going on in there are more important than others? Like, some of these differences don't make much of a difference. And then in the human brain, you can think about things like, you've got oscillations at different frequencies, and then you have to have nested oscillations of a certain kind, or phase locking, so that you'll get a population that's active, and then some other area will not be receiving that because they're being inhibited at that point in time, or the input's being inhibited. All kinds of things you do to make the system more predictable, in a sense. And you'll find a recurrent inhibition, so that the system won't respond to what's going on for a period of time after you've had some population of cells that were active. So I'm just wondering, if while you were going through this and determining that some things play more of a role than others, if you identified anything that you would call sort of systematic strategies that would make even these very different neurons, or all the shapes are slightly different, right? Make sure that those differences didn't have as much impact for a system that is basically hardwired and has to do a certain thing. Is there any kind of like encapsulating strategy how would you define that one thing makes more of a difference than the other?
I think one important to make sure that different, for example, to make sure that the structure of neurons, or the structure of a population doesn't can adjust to these differences, is a great example is plasticity between neurons, to make sure that it can adapt to different circumstances where it needs to have this specific voltage response. Intrinsically, there's also, when you move into other neurons in the brain, there are more variables involved. Sometimes since I have been working I have also been working in cortical function in human brain, or in mice. There are parameters such as self-inhibition of a neuron that make sure that the one particular part of a recurrent neural net in a neural population doesn't fire too much. It has states where the whole population is silent. So in itself, if there are different aspects in a circuit that can affect what type of how does a network respond to different things, and how does it adjust itself to make sure, for example, that it's not constrained to just extrinsic properties of a neuron.
One thing that very much helps in the fly is that the fly is physically small. And so the detailed structure of how many branches you have going exactly where doesn't matter much. It's like if you're thinking of it as the voltage, it's like filling the whole thing up, you're just lowering it and raising it. It's not like you have a long canal where the different levels can vary independently. It's all sort of one structure electrically. And so, at least on this scale, this is a small part of a small brain. And you wouldn't have that advantage in a mammal or a person. But it's one way the fly can be used to make sure the exact details aren't very important. It doesn't really matter where you connect to on that neuron. You can connect to a different spot, it would work the same. It doesn't really matter whether it has 10 branches or 9, as long as it has the same number of synapses. It's all that stuff sort of cancels out. And I think the fly uses that for sure to try to get the same response, despite individual variations of neurons, individual variations of things, and fly-to-fly variations. And the other thing it does that helps it a lot is when it's, the way it's using this is mostly for navigation. So it's trying to figure out which way it's facing. But to do that, it integrates over the whole field of view. Each of these circuits is just looking at a tiny patch, just a few pixels, say it's moving left or right. But it doesn't use that for navigation. Instead, it uses some global average. And that's much less sensitive than the individual circuits. And so I think it's using a lot of that too. Now, whether that's applicable to mammals, we have no idea.
All right. Let me just check quickly to see if we've got any online questions that came in, besides the one I already mentioned. I think that was that. And if there are no further questions in the room, then I think we'll thank Jonathan, and we'll take an 8-minute break before the next talk is due.
All right, thank you very much.
Thank you.